Let's review. The amino acid attached to a transfer RNA and the messenger RNA delivered by a shuttle protein meet together within this cluster of molecules called a ribosome. The ribosome has come together to perform an important service for Cell City. Before we learn what that is, let's move the transfer and messenger RNAs out of the way so that we can see what the ribosome is made of. Proteins form part of the ribosome, along with a third type of RNA called ribosomal RNA. Ribosomal RNA is composed of RNA nucleotides, the same four building blocks that make up messenger and transfer RNA. RNA nucleotides are molecules created within Cell City. Although their structure looks very similar, they each differ in ways that make them very useful. We'll learn more about nucleotides and their chemistry shortly. The ribosome, in reality, is a very complex molecular machine. The depiction you see here is a very simplified version. We've created it in this way to help you more easily understand how a ribosome works. See this chain of amino acids? We interrupted the ribosome just as it was beginning to make it. That's the ribosome's job, to create amino acid chains. We'll now return messenger RNA and transfer RNA to the ribosome and invite other tRNA amino acid complexes to drop in. Watch the ribosome as it continues its work. The ribosome catches a tRNA amino acid complex and holds it in place, while a ribosomal protein removes its amino acid and transfers it to the end of a growing amino acid chain. The amino acid chain is flexible, like a string of beads. The ribosome must have energy to perform its work. In fact, one of the reasons why you feed yourself is to supply Cell City with the raw materials to create ATP. ATP powers the ribosome. Now focus on the messenger RNA. Its entire length will pass through the ribosome 3 blocks at a time. The ribosome pauses upon each consecutive 3-block segment and allows a tRNA to land on it. As tRNA meets mRNA, the ribosome transfers the tRNA's amino acid to a growing amino acid chain. This match and transfer process will go on until the entire mRNA is read and the amino acid chain is completed. So what is messenger RNA? It is a bench offering seats along its entire length, 3 blocks long, one after another, upon which tRNAs sit and offer their amino acids to the ribosome. But each tRNA must remain seated long enough for the ribosome to harvest its amino acid. Chemical attraction between mRNA and tRNA allows this to occur. Here, a tRNA carrying the amino acid tyrosine has found an attractive seat. Interestingly, it is the only tRNA amino acid complex that finds this particular seat attractive. Other complexes carrying other amino acids do not. For every seat along mRNA's entire length, we find the same selectivity occurring. only one type of tRNA amino acid complex out of the many that exist will stick to a given seed. The consequences of this selective matching are very important, so let's take a closer look at the chemical nature of RNA. The 4 nucleotides that make up RNA are linked together in a chain. Their names are adenosine, cytidine, guanosine, and uridine. In this presentation, we depict each RNA nucleotide as a colored block, sometimes labeled with the first initial of its name. Nucleotides react with one another because of their differing chemical natures. For example, when U approaches A and G approaches C, they attract and stick to each other. Switch partners and the opposite occurs. G and A and U and C repel each other. So you can now see why this 3-nucleotide segment of mRNA binds so well to this 3-nucleotide segment of tRNA. Each nucleotide pairing between them is chemically attractive, or complementary. A's complementing U's, and G's complementing C's. On mRNA, this site is called a codon. On tRNA, this site is called the anticodon. If the anticodon of a tRNA is not complementary to a codon site, for example, if it offers an uncomplementary G to a U, or G to an A, or C to a C, it will be rejected. The ribosome makes it easier for the correct mRNA-tRNA matching to take place, codon after codon, down the entire mRNA line. But let's simplify the picture a bit to really understand what's going on. A messenger RNA, most simply defined, is a chain of nucleotides. But to tRNAs, it is a long bench of attractive codons, upon which they will stick if they possess the correct matching anticodons. If we dismiss the tRNAs for now, but leave their amino acids in view, we see that each 3-letter mRNA codon can be read as an amino acid. UUU, for example, translates into phenylalanine, GAA into glutamic acid, and AUG into methionine. A ribosome reading an mRNA is simply deciphering a list of 3-letter codons into a string of amino acids. Or, as scientists would say, it is translating the genetic code. To more clearly illustrate this, let's simplify the ribosome even further. Imagine the ribosome as a decoding machine, decoding mRNA into amino acid chains. If the mRNA code is long, the amino acid chain will be long. If short, the amino acid chain will be short. Observe that as mRNAs differ in their nucleotide sequences, the amino acid chains they code for differ in their amino acid sequences. From snack to digest, to the center of Cell City. We have been tracking the fate of the protein we eat and have discovered that food protein amino acids are reconnected in Cell City to create amino acid chains. As you might have already guessed, these chains become parts of protein citizens. We'll see how in the next presentation.